Hello everyone. Today we are going to start with the next point in the same topic, that is reproduction in lower and higher plants, that is development of endosperm and development of embryo. So before start this topic, let us take one small review of our previous lecture. In previous lecture, we discussed about the pollen pistil interaction, how the pollen deposited on the pistil then develops into pollen tube and it enters into the ovule, and what are the mechanism behind this that we discussed in previous lecture. Then we discussed about the artificial hybridization, which is significant in the crop improvement program, where the emasculation and bagging is there. And the most important point in last lecture we discussed, that is the double fertilization, where the fertilization takes place twice, first and second fertilization. So the reference of this fertilization, first and second, or the result of first and second fertilization, that is the development of embryo and development of endosperm respectively, that we are going to study in this lecture, that is the development of endosperm and development of embryo. We all know that in a double fertilization process, the development of embryo, it is the result of first fertilization, that is syngamy, where first male gamete fuses with egg, which gives zygote, which is diploid, and further that zygote develops into embryo. And the result of second fertilization or triple fusion is second male gamete fuses with diploid secondary nucleus, which gives rise to the primary endosperm nucleus, which further develops into embryo. So in this lecture we are going to start with the development of endosperm. What is endosperm? The endosperm, it develops by the second fertilization, where the fusion of second male gamete with secondary nucleus results into the formation of primary endosperm nucleus, which is triploid primary endosperm nucleus, when repeatedly divides by mitotic division to form the nutritive tissue, is called endosperm. So the endosperm formation, it is the result of second fertilization, where the triploid primary endosperm nucleus divides repetitive mitotic divisions to form nutritive tissue, is called endosperm. So the formation of endosperm, it is the result of post fertilization, after fertilization, that is the fusion of male and female gamete, result of post fertilization. So therefore after the post fertilization, the ovule, embryo and endosperm, which develops simultaneously from the in the ovule, that is the development of embryo and endosperm, it takes simultaneously, and on the basis of this development, that is the mode of development, the endosperms are of three types. There are three types of endosperm, namely first one, that is the nuclear endosperm, second one, that is the cellular endosperm, and third one, that is the halobial endosperm. One more type of endosperm is there, that is the mosaic endosperm, that we are going to study later. So start the first type of endosperm, that is the nuclear endosperm. The nuclear endosperm, it is the most common type of endosperm, which is found in 161 angiospermic families. Most common type in among the endosperm type. It is the most common type of endosperm which is found in 161 angiospermic families. Then how the development of nuclear type of endosperm takes place? In a nuclear type of endosperm, if we are going to study the diagram, here is diagram of nuclear type of endosperm. Here one, that is the zygote, which is also called as the oosphere. In a nuclear type of endosperm, the primary endosperm nucleus, divides mitotically without wall formation. हे अतिशय महत्वाचं आहे इथं. The triploid primary endosperm nucleus divides mitotically without wall formation to produce large number of free nuclei. And one more important feature in this nuclear type of endosperm, here it is in second diagram. At the center there is a presence of big vacuole. So this big vacuole which is at the center, it pushes the nuclei towards the periphery. बाहेरच्या बाजूला पेरिफेरल साइडला हा मोठा जो काही व्हॅक्यूल आहे हा त्या न्यूक्लियाना काय करत असतो, पुश करत असतो. अँड लॅटर अराउंड these free nuclei there is formation of wall. Here there is wall formation around these nuclei takes place. Hence, the multicellular endosperm is formed. But in several cases, cell wall formation is incomplete. Examples where the nuclear type of endosperm is present, for example, wheat, sunflower and coconut. Here the nuclear type of endosperm is present. In case of coconut, has multicellular endosperm in the outer part and free nuclear as well as vacuolated endosperm in the center. Then let us start with the second type of endosperm, that is the cellular endosperm. In case of cellular endosperm, again the same. The triploid primary endosperm nucleus divides mitotically, but immediately it is followed by the wall formation. In case of nuclear endosperm, there is no wall formation after the mitotic division. But in case of cellular endosperm, after the mitotic division in a triploid primary endosperm nucleus, there is wall formation takes place. Therefore, the endosperm is cellular right from the beginning. And this type of cellular endosperm, it is mostly observed in 72 families in dicotyledonous plants. For example, balsam, petunia, adoxa. These are the examples of the plants where the cellular type of the endosperm is present. The major difference between the nuclear and cellular is both they are having the mitotic division in relation with the triploid primary endosperm, but in case of nuclear there is no wall formation after the mitotic division, but in case of cellular type immediately there is wall formation around the triploid primary endosperm nucleus after the mitotic division. Then next one, that is the halobial type of endosperm. The halobial word, it comes from the halobial series in monocotyledonous plant. That means this type of endosperm, it is found only in monocotyledonous plant, but in that monocotyledonous plant the series, halobial series, it shows this type of the halobial type of the endosperm. What happened in halobial type of the endosperm? Here in case of halobial type of endosperm, the first division of a primary endosperm nucleus, it is followed by this transverse wall formations which divides this cell unequally. One there is larger part, another one that is the smaller part. The smaller part or the smaller cell, it is called as the chalazal cell, and the larger cell, it is called as the micropylar cell. And in both, in this larger cell, that is the micropylar cell, in this smaller cell, that is the chalazal cell, the nuclei in each cell divides free nuclear division, and then only there is wall formation between these nuclei takes place. Therefore, the halobial endosperm, it is the intermediate type of endosperm in between the nuclear and cellular type of the endosperm. For example, this type of endosperm, it is found in the plant Asphodelus. So these are the three basic types of the endosperm. One more type of the endosperm, that is the mosaic endosperm. Here this mosaic endosperm, we know this seed, it is familiar to us, that is the corn seed or the maize seed. Here this endosperm, clearly it shows these two distinct patches of the color, and it is the identity or the mosaic pattern. Therefore, the name is given as the mosaic endosperm. In mosaic endosperm, that endosperm containing the tissues of different types. Two different types of the tissues are present. For example, in the plants like corn, endosperm containing the patches of two different color. Here there is one color, at the center there is one color. So these two distinct patches, it looks like the mosaic pattern. Therefore the name for the endosperm is mosaic endosperm. So these are the three basic types of the endosperm, that is the nuclear endosperm, cellular endosperm and halobial endosperm, which are based on the mode of the development of the endosperm, and this one, that is the mosaic endosperm. So it is about the development of endosperm. The long answer question may be asked on this content, that is the describe the various types of the endosperm in angiospermic plant. So diagrams are also essential to draw to write the answer of this question, or separate question on each type of endosperm it may be asked, like the describe nuclear type of the endosperm separately, cellular type of the endosperm separately, allovial type of the endosperm separately, or the mosaic type of the endosperm separately. So it is all about the development of endosperm, and we know that endosperm, it is the nutritive tissue. Then come towards the development of embryo. Embryo development, this embryo, it is the result of first fertilization or syngamy, where the fusion of first male gamete with the egg, which gives rise to the zygote, and that zygote further gives rise to the embryo. So what is embryo, or the formation of embryo? It is the embryogenesis. What is embryogenesis? It is the process of development of zygote into embryo. It is called as the embryogenesis. So this embryo formation process from the zygote, it is at the ovule, but in the region of that ovule, it is called as the micropylar end. There is chalazal end also and micropylar end also. So the embryo is developed at the micropylar end of the ovule, and this development of embryo, it is by the different divisions. So at the base, in the development of embryo is zygotes. So this zygote first divides into two-celled structure, that is the proembryo, and in this two-celled structure, that is proembryo, it consists of two-celled structure, one that is the larger cell, another one that is the smaller cell. The larger cell, it is towards the micropylar side. It is called the basal or suspensor initial cell, and smaller cell, it is called the chalazal or the terminal or embryonal initial cell. So this development of embryo, let us know the development of embryo by this diagrammatic representation. So here it is the zygote or oosphere. Here it is zygote or oosphere. हे नीट समजून घ्या. आणि हा जो काही A, B, C, D, E, F, G, H ह्या सिक्वेन्स मध्ये आपल्याला या ठिकाणी जो काही एम्ब्रियो आहे हा एम्ब्रियो इथे डेव्हलप झालेला दिसतो. हा कसा होतो तर सुरुवातीला काय होतं, द झायगोट और oosphere डिवाइड्स इनटू टू सेल्ड स्ट्रक्चर. आता हे पाहू शकतो आपण, इथे एक सेल आहे, इथे एक सेल आहे. आता ह्या टू सेल्ड स्ट्रक्चरला काय बोललेलं आहे, प्रो एम्ब्रियो. प्रो एम्ब्रियो. आता यापैकी लार्जर एक आहे आणि यापैकी स्मॉलर आहे. द लार्जर सेल ही वरच्या बाजूची, the larger cell which is present towards the micropyle, it is called as the basal or suspensor initial cell. हे नीट समजून घ्या. लार्जर सेल आहे आणि एक स्मॉलर सेल आहे. the larger cell which is towards the micropyle called as the basal or suspensor initial, and the smaller cell towards the chalazal side, it is called as the terminal or embryonal initial. And the next development of embryo, it takes place in this suspensor initial cell and in this embryonal initial cell. Then what happened in the suspensor cell and what happened in embryonal initial cell? Here that suspensor initial cell in diagrams, suspensor cell, this suspensor cell divides transversely to produce the filamentous suspensor cell of 6 to 10 cells. And these 6 to 10 cells, here these 6 to 10 cells, the first cell of suspensor cell towards the micropylar end, it becomes swollen and it functions as the hostorium. The first cell which is present at the micropylar end, it becomes swollen and it functions as a hostorium. And this lowermost cell of this suspensor is called as the hypophysis. Is called as the hypophysis. So what is the importance of this hostorium or this hypophysis? That suspensor cell or hostorium or hypophysis, it pushes the embryo into the endosperm for the nutrition. And what happened with this embryonal initial cell? That embryonal initial cell, आता इथून खालचा जो सगळा पोर्शन आहे तो embryonal initial cells आहे. This embryonal initial cell, it undergoes mitotic division to form the octant. Here this structure togetherly called as the octant. Octant means 8, 4 plus 4. So the lower tier of these 4 cells, here 4 खालच्या cell, the lower tier of these 4 cells of the octant, it gives rise to the hypocotyle and radical. It gives rise to the hypocotyle and radical. आता हा जो काही व्ह्यू आहे तो आपल्याला इथे एनलार्ज केलेला दिसतो पहा. Here that hypocotyle and radical, it is formed by the lower tier of the 4 cells of the octant. Then upper tier of the 4 cells, it gives rise to the plumule. Here it is plumule and 1 or 2 cotyledon. 1 or 2, it depends on the plant, either it is monocotyledonous or dicotyledonous. So the development of this octant stage, up to the development of this octant stage in both dicot and monocot, the development of embryo is common. When it is monocot plant, there is formation of single cotyledon. If it is dicotyledon plant, then there is formation of two cotyledons. Then come towards this diagram, that is H. Here in this diagram, that embryonal axis which are called as the plumule and radicle. Here it is radicle. This radicle which further gives rise to the root. That radicle is covered by the protective sheath. It is called as the coleorhiza. Coleorhiza. And another type of the embryonal axis, that is the plumule. Here there is plumule, and that plumule it is covered by the protective sheath. It is called as the coleotile. Coleotile. So these are the two protective sheaths on the embryonal axis, that is the coleoptile which covers the plumule and coleorhiza which covers the radicle. So plumule and radicle, these are the two protective to embryonal axis which is protected by coleoptile and coleorhiza. And this structure scutellum, it is the single shield shaped cotyledon which covers the cotyledon, maybe monocotyledon or dicotyledon. And after this development of embryo, the ovule it develops into the seed and the ovary develops into the fruits. So here there is development of dicot embryo in capsula. These are the same diagrams like these diagrams in monocot, and here it is dicot. The features are given in these figures. So at the end, the development of embryo takes place by the different stages. Proembryo, then it divides into two cellular stage, larger cell, suspensor initial cell, smaller cell, embryonal initial cell, and at the end the development of embryo takes place. The long answer question on this content may be asked like the describe the development of embryo. Again, give the functions of the plumule, give the functions of radicle, what are the protective covering, what is the role of suspensor initial cell, embryonal initial cell, what is the role of hostorium, hypophysis, what is octant stage. All these questions may be asked on this content. So be prepared with this video and continue your study. Thank you.